Hi, everyone. I'm Dr. Nathan Babcock, and it's a pleasure for me to present today at the Resonance Symposium in São Paulo, Brazil, on my work on quantum biology for health and medicine. To begin, I'd like to provide a brief history of quantum physics, given in terms of the technology that has enabled over the years. Going back to 1911, fluorescence microscopy has allowed us to look into cells using fluorescent dyes to show the various parts of living cells. In the picture we see here, the nucleus of cells stained in blue, with the cytoskeleton shown in green and the actin filaments shown in orange color. By 1933, electron microscopy enabled us to look deeper into the living world. We can see the fine resolution of a number of grains of pollen here. And by 1947, the semiconductor transistor has enabled microchips and all the wonderful computing technology that we see today. By 1958, the laser was invented, which is now very common. Laser pointers we see in many in-person presentations. And by 1977, magnetic resonance, technology that's often used in health applications to look into the body. And with functional magnetic resonance imaging today, we can often even see life at work. Currently, quantum computing offers a wealth of possible applications, both in terms of studying healthcare data and also in terms of understanding various aspects of chemistry, molecular biology, and as I'll discuss with you today, quantum biology. But to begin, we need to know a bit about quanta, the actual quantum particles and waves of light and matter. The photon is a single massless particle of light. It's often represented in diagrams as kind of a squiggly line, and it carries energy that's proportional to its frequency. In an equation shown here, where E is the energy, F is the frequency, and H is Planck's constant, the fundamental constant of quantum physics. A photon can interact with an electron, a tiny negatively charged particle. These electrons are typically found orbiting the nuclei of atoms, which contain protons, a positively charged particle, 1,000 or more times heavier than an electron, and neutrons are also found in the nucleus of an atom, which is often represented in a familiar picture where the electrons orbit the nucleus, although the nucleus is in fact much smaller than is revealed in this picture. Now, when an electron absorbs a photon of light, it goes from the lower energy state to the excited state, absorbing a unit of energy that is again proportional to the frequency of light. This excited state can relax as it drops down to the ground state of the electron, releasing the energy as another photon. Electrons and other particles also have a property called spin, which is the intrinsic angular momentum of a particle. It's kind of like a spinning top if that top could never stop spinning. As the intrinsic angular momentum, the electron cannot be without it. And it is quantized such that the spin can only point in one of two directions along any axis we observe it along, for example, up or down, left or right, forward or back, for an electron, at least. There are other particles with other properties, according to the standard model of particle physics, but for most biological applications, understanding the photon, the electron, proton, and neutron inside the nucleus of the atom, and the unique property of spin are often more than adequate. As an introduction to quantum biology, it's worth noting that quantum biology is the study of biological processes that cannot be predicted or adequately described using only the principles of classical physics. Quantum effects in biology include things like photosynthesis, electron tunneling during cellular respiration, and many fundamental aspects of molecular structure. Quantum biology research has important implications for models of health and medicine, with a variety of applications that I'll discuss today. And it is essential, ultimately, to have an appreciation for quantum mechanics in order to understand the fundamental principles of life. Now, electromagnetic modulation is an important aspect of cell metabolism and can be relevant to many processes, disease processes, including the formation of tumors. Now, inside cells, I showed the picture of the microtubules in the cytoskeleton surrounding the nucleus. They were shown in green, and they connect with the mitochondria, the so-called energy powerhouses of the cell, to produce electrical oscillations in the very polar, positively and negatively charged environment of the inside of the cell matrix. Now, these kind of dynamics are fundamental to many health processes, including disease processes like tumorigenesis, the formation of tumors, and they can be driven by the microtubule cytoskeleton in a number of complex processes. So mitochondria are implicated in all cancer-related processes and also in many other health and disease-related processes as the primary energy producers in the cell. So there's more I could say about the relationship between energy transduction and a disease process like cancer. But I'd like to take a moment to talk a bit about the formation of excited states in microtubules. Now, ROS, those are reactive oxygen species, ROS, are produced in mitochondria, and they're a normal part of cellular physiology that can also produce very weak emissions of light, ultra-weak photon emissions in cells. Now, the formation of reactive oxygen-induced excitonic states can, in principle, propagate through quantum transfer in the microtubules. And these microtubule networks may transport reactive oxygen species-generated photon energy for cell signals. And I say this to give you some impression of where we're going as we continue, the role for reactive oxygen species and microtubules can control many health and disease aspects of the cell. Microtubules are sensory motor integrators. They combine information, sensing, and motor information, movement information in the cell. In fact, they can sense structural tension, stress related to the surrounding environment, pressure, the humidity, redox states, the chemical states of the cell, and even light. Now, microtubules are not static structures. They require energy that is imparted to them in a process called GTP hydrolysis. GTP, guanosine triphosphate, is essentially the energy source for the microtubule cytoskeleton. And various processes require pumping through this energy source that enables a variety of coherent motions and potentially optical activity, and in fact, interactions between the mechanical states and chemical and optical states of the microtubules themselves. Why is this significant? If we look back at our picture of the cells that I showed early on, it's important because microtubules, as the main structural component of the cell, control not only structure, but cell division, cell motility for cells that move around, such as a white blood cell or any cell with movement capabilities. And as I mentioned, sensing. They're important stress sensors in the cell. They respond to mechanical tension. They enact proprioceptive control. That's control of the sensory environment of the cell, a cell's sense of what it's doing, its current status, and its own cellular perception of its morphology, its shape. Of course, the shape of the cell is very important to its own kind of inner sensation, inner knowledge, if you will, of itself. Microtubules are also good light harvesters, which shows that they have an important role to play not only in terms of the chemical and mechanical environment, but also the optical environment of the cell. Now, this is all quite significant when we begin to think about electromagnetic therapies, past and present. Number of therapies have been very successful and have led to, in fact, quite prestigious awards, such as the Nobel Prize in Medicine, which was awarded in 1903 to Nielsen Fins for his treatment of lupus vulgaris. That's a form of skin tuberculosis that he was able to treat successfully in 1893 with UV light. He was also able to treat smallpox with red light. Now, since then, UV phototherapy has become quite important for treating other conditions like plaque psoriasis, unpleasant skin condition that forms lesions on the hands. UV light treatments, either out in the sun or in specialized UV booths at a hospital, for example, can be very effective in controlling psoriasis. Electric currents have been used to enhance the rate of bone healing, and this is a well-known phenomena approved by the FDA in the US, has been used for several decades now, in which either electrical stimulation or magnetic stimulation is used to speed the recovery time of bone fracture. This all leads to the idea of what's called photobiomodulation, sometimes known as low-level laser therapy or infrared therapy, which has been shown to provide a number of benefits in treatments over the years as well. So there's no shall we say, medicine is no stranger to electromagnetic kind of therapies, but they're often not well understood and frequently underutilized because of the lack of understanding of the underlying mechanisms, even though, in fact, many of these therapies have been proven for decades, if not over a century now. So, where are we going with this? We're hoping that in the future, or even the near future, techniques like photobiomodulation, the control or modulation of biological effects through light, can be used for tissue repair, inflammation reduction, and analgesia, pain relief. A number of various conditions have already been shown to respond well to photobiomodulation, including osteoarthritis, bone healing as well, wound healing, and as suggested here, inflammatory conditions. A number of works have been done on mouse models in order to improve or understand the effect better. And these include the use of photobiomodulation, in conjunction with gold nanoparticles, which can create a resonance effect to increase wound healing, increase wound contraction, decrease inflammation, and decrease oxidative stress, while improving growth in a number of studies. As well, photobiomodulation therapy has been proven to improve the acute inflammatory response in mice. Now, there's some belief that this may be involved in a mechanism that relates to the electron transport chain in the mitochondria of the cell. And some studies have also showed that cannabinoid receptors and possibly ATP-sensitive potassium ion channels may be important in the response to these kind of treatments, as they can modulate pro and anti-inflammatory cytokines. Of course, cytokines became well known to the general knowledge at the emergence of the recent COVID pandemic and the fearful cytokine storm. So there's some research that have done to show that this kind of therapy could also be helpful for those kind of cytokine or inflammatory related diseases as well. Now, these kind of therapies work by modulating metabolism. For example, therapeutic nuclear magnetic resonance therapy can be used to persistently modify cell metabolism. In these kind of treatments, nuclear magnetic resonance is used not just to image cell, but to actually provide treatment through the interaction with the magnetic field and the nuclear spins in the cells. This can control hypoxic signaling, that is the cell's internal sense of how much oxygen it has to breathe, and also has an influence on the cell clock or circadian rhythm. Now, what is interesting is that these studies have shown that these kind of mechanisms can control the reactive oxygen species metabolism in the cell. And if cells are cultured under hypoxic, that is low oxygen conditions, in the presence of therapeutic NMR, that it can reduce glycolysis in the cell. Another relevant feature, if you're interested in something like cancer, where glycolysis is a key factor in the disease process. Interestingly enough, it increases reactive oxygen species in the mitochondrial respiration while increasing reactive oxygen species outside the cell, but decreasing reactive oxygen species, as I mentioned, along with glycolysis inside the cell. But what is really the most interesting here is not only can these preliminary studies show that this can enhance healing, but we really are beginning to see in-depth control of the cell metabolism itself, which in principle can open whole new avenues for medicine. We don't just treat the cells, we can actually enable them to heal faster on their own. Now, quantum electrodynamics, the interaction of light and matter, can already play an important role in diagnostics and what are called theranostics, therapy combined with diagnostics. As we saw a moment ago, the idea that NMR can be used not just for imaging, for diagnostics, but also as a therapy. Quantum sensing is being used to detect subtle changes in the cellular environment, and it can help provide advanced detection factors for driving and controlling things like cancer. In these kind of systems, we have a nanoparticle, in this case, an iron selenium nanoparticle, which is functionalized with organic molecules coated on its outside. It is optically active, and so we can, as I showed earlier, use light to excite it to an excited state. And then depending on the frequency of light we use, we might find different kind of outputs. But ultimately, the goal is to produce a fluorescent signal that allows us to see the actual effect. So what we have here, we begin by encapsulating the nanoparticle. We add, for example, a particular antibody using a number of typical biological molecules, and then they allow us to look inside the cell. So if we have cells that are stained using standard fluorescence techniques, we could see a significant difference in the lower right-hand corner with cells that have now have the inner characteristics being revealed through these antibody sensitive nanoparticles that actually seek out specific parts of the cell and bind to them depending on the antibody coating. Now, this is exciting because it allows us not just to image cells with a new level of precision, but if the nanoparticles additionally have specific features that allow them to not just emit light, but to heat up, emitting heat, or engage in other chemical processes, sometimes called chemodynamic therapy, we can now use the nanoparticles themselves to produce different oxidation states, produce heat, or even control other aspects of the cell chemistry, interacting with them through the control of electromagnetic radiation. Now, thinking even deeper into this kind of picture of electromagnetic control of parts of the cell or various nanoparticles, even enzymes injected into the cell, proof of principle demonstrations have showed control enzymatic DNA repair in human cells. UVB-mediated DNA damage has been reduced by treating cells with liposomes containing the DNA repair enzyme photolyase following exposure to photoreacting light. Now, if we see the photolyase molecule taken from the protein data bank, it binds to DNA and the various light-sensitive factors such as 8-HTF, tryptophan molecules, and the main cofactor, flavin adenine dinucleotide, can absorb light and use the energy from that light to repair damaged DNA. Human epithelial HeLa cells had been damaged by UV light in the study in vitro. The cells were treated with photolyase from photosynthetic bacteria, a completely different organism. The enzymes were activated using light, and they were shown to reduce apoptosis, cell death caused by DNA damage, and enhance DNA repair and surface receptor suppression. Another method work additively to show that in principle, this kind of DNA treatment could be combined with other treatments to get a concerted effect. And finally, as a control, it was shown that photolyase reduced apoptosis significantly compared to treatment of liposomes that did not contain the DNA repair enzyme. So this kind of treatment, really has great potential if we can imagine actually using enzymes that we can inject into the cell, or even providing photobiomodulation of existing enzymes in the cell to speed healing or change the underlying processes that might control disease states. This is a wonderful example because the magnetic field strength and even the direction of the magnetic field have been shown to be able to control DNA repair, and that a full characterization of the complete photocycle of this enzyme exists. So if we're interested in the research, there's a long way we can go developing this kind of therapy, at least in principle. Electromagnetic control of cellular metabolism has been shown in a number of cases. In fact, the real-time control of neutrophils, that is white blood cells, by very weak, low-frequency pulsed magnetic fields has revealed that we can actually control various immunological effects. As well, ultraviolet, visible, near-infrared light, ionizing radiation, and chromophore-assisted light inactivation. That was the therapy I mentioned on the previous slide with the nanoparticles, where we can actually use light to activate a nanoparticle, which then can release heat or some kind of chemical effect to kill a cancer cell, or engage in any other kind of potential redox control in the cell that can affect, for example, the actin cytoskeleton of the cell, with implications for cell function, motility, and again, the cell shape, the morphology. So we know that UV radiation, visible and near infrared radiation, even X-ray radiation can have an influence on the reactive oxygen species that control things like the cytoskeleton, both through the microtubules and the actin network, which has interesting implications for medicine and health biology in general. Now, reactive oxygen species has also been able to show to modulate microtubule ordering, revealing mechanistic insight into reactive oxygen species homeostasis that in turn controls the actual shape of the cell. Here we see two different flower petal cells, one which has been treated with reactive oxygen species to make it more bulbous, whereas the control example on the left shows the typical cone cell without this kind of reactive oxygen species treatment. Again, if we can use light or magnetic fields to control the reactive oxygen species in the cell, we have an enormous amount of control even over such things as the shape of the cell. So the list goes on and on. Magnetic control of cellular regeneration has been shown that weak magnetic fields can be used to selectively enhance or disrupt stem cell mediated healing in small invertebrates. So these organisms, we've shown that whether they're treated with weak magnetic fields or not, can control their rate of healing if they're injured, in fact, cut apart and allowed to regrow. And we see that different magnetic field strengths can, in fact, produce different healing outcomes, exhibiting a wide range of control of this kind of recovery process in this organism that is completely dictated by the magnetic field effect. Now, it's believed that these interact with cells by controlling, again, the reactive oxygen species accumulation in the cell. And so, if we imagine a particular molecule that dissociates it produces two different spin states of the electronic state. And what is believed is that the magnetic field actually affects electron spin flip rates, so that it's actually control of the quantum states of the spin that in principle, and it would appear in practice, is controlling the underlying rate of healing in these simple organisms. Now, what kind of reaction could this be controlling? Well, without getting into a lot of the details, we can consider a molecule like oxygen. Here are, from left to right, different redox states of oxygen, excitation states of oxygen, ground state oxygen, the lowest energy state on the left, and then singlet oxygen, which is known to be a reactive state, a superoxide radical, another reactive radical, and a peroxide ion on the far right. What is interesting is that the singlet oxygen has two different states it can enter into. We also look at a kind of activated state of singlet oxygen, where the two electrons that are shown as the arrows now are in different energy levels, shown as sigma 2p, pi 2p, pi star 2p, and sigma star 2p. Without getting into this spectroscopy notation, what is important that the electrons, in fact, the electron spins that are shown as these arrows pointing either up or down, can in principle be controlled in molecules with open shell configurations like oxygen, in principle, which would appear to govern healing processes in organisms like the simple invertebrate shown on the previous slide, but in fact could be very relevant to even higher forms of organisms. Well, what does this all mean? Electromagnetic control of health, disease, healing. Well, it's not all good news, because significant associations have been observed between exposure to extremely low frequency magnetic fields and childhood leukemia. Furthermore, even in this case, a possible dose response effect was observed. So I wouldn't just go wire yourself to some electromagnetic fields, because it's not necessarily going to be a positive effect. Earth strength magnetic fields do appear to be important for health and vitality, though, and the deprivation of an organism from the Earth's magnetic field, if we actually shield an organism from the healthy, natural magnetic field of the Earth, can cause potentially damage, or certainly can reduce neurogenesis and cognition. In some organisms, it was shown to influence the feeding patterns, disrupting the appetite of particular migratory insects. And it has even been shown that tubulin assembly into microtubules is disordered in the hypogeomagnetic field, the magnetic field weaker than that of the Earth, which is already quite small. So it appears that magnetic fields are very important, but in the right dose, and it would appear even at the right frequency. So what are we able to do? And this study from 2018 results highlight the ability of extremely low frequency magnetic fields to modulate inflammation and may play an important role in wound repair. So I ask, what is a doctor or therapist to do? It seems that these kind of magnetic fields have a great potential to enable healing processes. They seem, in fact, important in terms of the Earth's magnetic field to maintain healthy biological processes to begin with, but they seem to have what is called a double-edged sword, that they may or may not be beneficial depending on the underlying dose. So it's very important to know, ultimately what is going on with these mechanisms to be ensured that we are maximizing the health benefits of potential treatments and minimizing potential health risks, which may not be negligible. So, in conclusion, we have the emergence of a coherent picture of electromagnetics and dynamics for disease. That electromagnetic and light-based therapies have a long history of medical success, and phototherapies and resonance effects can control disease-related metabolic factors. These include inflammation, dysregulated healing patterns, pain, This includes redox dyshomeostasis, the actual dysfunction of the redox state of the cell, and as well immune responses. Now, nanoparticle quantum sensors can be used to image, diagnose, and treat diseases through a process called nanotheranostics. That is nano-sized therapy and diagnostics rolled into one. And these kind of potential treatments using nanoparticles are probably one of the most promising near-term treatment based on distinct quantum and even electrodynamic effects in the cell. Now, we know through research that electromagnetic fields can control enzymatic processes that govern things like healing and DNA repair. And electromagnetic fields are intimately connected to microtubules and mitochondria in processes that control health and illness. These include energetics, redox signaling, and morphology. Now, what is most interesting to me is that cells, through the microtubule network, through the mitochondrial networks, can integrate chemical, electrical, magnetic, mechanical, and it is increasingly being shown even optical processes. And it's important to integrate these processes in a coherent, cohesive way so that the cell has a meaningful understanding of its environment as it searches for food, avoids danger, or in a complex multicellular organism cooperates with its surrounding cells and doesn't resort to a disease state that could potentially damage other cells. This overall paints a picture where we need an in-depth, microscopic, even nanoscopic picture of the cellular environment where various interactions between light and matter, electromagnetic fields, and the underlying pigments and other light-sensitive molecules that are shown to interact at this scale can function in an appropriate way. And potentially, with the correct kind of knowledge and understanding of the underlying mechanisms, we may be able to control these processes. In fact, we've shown the control of these processes. It's just a matter of whether we can develop the underlying principles and applications into actual research-based medicine. So I'm very happy to have presented to you today. And if you have any questions, my name is Dr. Nathan S. Babcock. I've put my email on the slide. It's been wonderful speaking with you today, and I hope that we can be in contact. I look forward to taking any questions you may have about quantum biology in general, its role in physiology, cellular biology, and various aspects of cellular health, which, as I said, begin with the overall structure, the shape of the cell, include its redox state, the homeostatic state of the chemistry of the cell, and can even include potentially the cell's interaction with its light environment, interaction with other cells, and its overall state of health, which is crucial for various chronic diseases that continue to plague humanity and are not often treated using medicine of the conventional sense, but may require a more in-depth understanding of the actual cellular environment on the quantum level in order to appreciate the many underlying factors that go into controlling these processes. So thank you again very much. I'm very grateful to our sponsor, the Resonance Symposium, and hosts in São Paulo, Brazil. I thank my previous collaborators at Howard University, where some of my most recent research was performed. And I'm grateful for all of your attention, and I look forward to talking with you in the future. Thanks again so much, and have a good conference.